So it is my pleasure to introduce Alex and Guillaume. They're at Mila and the University of Montreal, and today they're going to be talking about flow matching and conditional protein backbone generation with their new methods that are out. So please help me welcome them to give their talk today. Thank you so much for being with us, Alex and Guillaume.
Cool. Yeah, thanks so much for having us. Yeah, so today we're going to be talking about these flow matching models for protein design that we've been working on. So a little bit about the first one, and then Guillaume will talk more about the newer version. Yeah, so just an outline. We're going to talk about sort of problem setup, flow matching, and the first FoldFlow, and then FoldFlow 2, and some experiments, and then finally sort of what we're doing, what we're interested in next. Cool. So just for everybody, I think most people know what proteins are here, but just a summary for our purposes, proteins are sort of large, complex molecules, and they're very important in living organisms. And what's very interesting, of course, is they're composed of many smaller units, which are amino acids, and they're linked together and make a specific chain. And this is really the important part here. And we're trying to figure out 3D structure. So linking sequence and structure is a big part of this. And so these two pieces are critical. So the two main modalities, sequence and structure. So just thinking about the representation here, you have a sort of a sequence representation, which is, well, represented by which residue, which kind of residue each atom is, and then sort of the 3D positions where they are in 3D space. Yeah, so this is sort of the basics, but the sort of landscape here is, well, what do you want to do with these, right? So there's many sorts of different models that you can train. So the sort of the ones that everybody knows about is the folding models. So trying to go from sequence to structure. This is a big task. ESM fold and AlphaFold 2 and really many other models are trying to go from sequence to structure, trying to understand the sort of how sequence determines structure, at least in crystal form. The second thing that's been sort of somewhat more recent was inverse folding, so trying to determine, given a structure, what is the possible sequence. And the sort of big models in the space are protein MPNN and ESM inverse folding. So this takes a 3D structure, 3D coordinates, and tries to find a sequence that fits that structure that will sort of refold in that structure. And then so what we're going to mostly talk about today is these models that go from structure to structure. So mostly a noisy structure to a cleaner structure. And so this allows you to generate new structures and potentially conditional generation of structures. So that's the important part here. And the final type of model is sort of sequence to sequence models, right? Language models. I think you guys heard about it last week with EMSIM3, but which can also do other things, but of course is mainly sequence to sequence. And so these are sort of the interesting first generation models, and we'll talk about sort of what's coming up shortly. So sort of the protein task. So the first task is unconditional generation. So this is the main task we focused on, and others focused on is sort of, can you generate new and good proteins? So just from a noisy structure, can you generate a structure that looks like a protein that could be made in real life? The second task would be motif scaffolding. So trying to understand, well, given part of a structure, how can you scaffold it to be in that shape? And so this is sort of a in painting, if you're thinking about machine learning, so you have a fixed part and a designed part and trying to make something that sort of folds together well. And the final piece is going to be binder design. So trying to fix a target and then design a separate protein that binds to it. So very similar, of course, this is in painting, but sort of for multimers. So these are sort of the three things that we're most interested, I would say. Yeah, so I guess the way to think about these equivariances. So proteins, right, exist in 3D space, but of course, if you rotate a protein or move it around, it's still the same protein. And so this is sort of the space of SE3. So that means equivariant to translations and rotations. And there's sort of two ways to enforce this. This is either directly building it in the model or doing data augmentation. So for instance, the most recent AlphaFold 3 does it mostly through data augmentation. But most of the models we've been working on build it directly into the model. So sort of the first deep learning folding models built it directly in. And maybe the more recent way for AF3 is still exploring, right, is the sort of data augmentation strategy. So we're looking at sort of how do you model directly on SE3 and trying to say, well, translations and rotations don't really matter that much. Cool. Okay, so yeah, FoldFlow is sort of SE3 is flow matching on SE3, and I'll tell you a little bit about flow matching in a second. But the basic idea is, yeah, so this is a unique representation that's sort of translation and rotation invariant. And that says, right, we're just looking at the backbone. So the sort of, we're ignoring side chains and just looking at the backbone and saying the SE3 part is sort of for each amino acid, where is it positioned and how is it rotated in space. So I sort of think about it as sort of a chain of like a necklace with each orientation is sort of how the chain hangs off of it. So that's sort of how the representation is going to work. And then of course we need to do flow matching in this space. So that's sort of the next piece. Yeah, so right, this is sort of n amino acids, and each one is a frame, so the rotation and translation. And you have sort of these fixed coordinates with relative distances and angles. And so what you're really looking for is sort of where the C alpha atom is, and then how are the rest of the atoms rotated around it. And so the sort of rotation is the SO3, so the rotation space, and the translation is denoted by SI here. And so, yeah, we're looking for sort of this movement in the space and flowing from sort of noise coordinates, a noisy translation and rotation, to sort of the true translation and rotation. Great, yeah, and so this is basically how you apply this sort of SE3 transformation to these frames. Cool. Yeah, so now I'll talk a little bit about flow matching. So yeah, I've been working in this space for a while. I guess sort of why you should care about flow matching. I think flow matching is sort of very related to diffusion, but there's a couple advantages here. So it's sort of more general, and I'll tell you exactly why. But the big things that you are really interested in is sort of how do you speed up training with a different objective? How can you get faster inference? And sort of yeah, we're going to use optimal transport here. And then finally is sort of how do you implement these with sort of on rotation and translation space. So showing that flows are sort of easier to implement than diffusion on manifolds. So these are sort of three important things for flow matching. And we're going to take this score matching loss, which basically says at every point in space and time, train a network to match your conditional score at every place. The exact same thing holds for flows. So it turns out that basically for any vector field that this math works out. And so that's a general idea here is we're going to take this sort of score matching loss, which is very effective in fusion, and then sort of generalize it. And it gives you sort of ways to play with the objective to make it faster for training, faster for inference, and also allows you to not have to do some sort of heat diffusion on manifolds, which is very challenging. So I like this picture sort of in the bottom right from Lippmann is sort of you can choose your paths basically. So this says for a data point right here, you're sort of looking at the paths back to a normal zone or one Gaussian. And diffusion can have very curvy paths where if you pick the right kind of paths, and you get very straight lines. And straight lines are really good for fast inference. So sort of allowing you to go from a score matching framework to a flow matching framework allows you to sort of make choices like which paths would you want to take for faster inference and sort of lower variance for faster training. Yeah, so diffusion models, right? Just a little reminder how this works is you take something clean, some data points, and then you add noise to corrupt it, and then you try and learn sort of the reverse function. And this reversing function is related to the score, basically. So you want to learn basically which direction is your data at every point. And so this allows you to train a function that approximates your score, and then so it takes in your current place, outputs your score, and then you can use this equation to sort of denoise your function. So at every step, you sort of walk in the direction of your data a little bit, and then add a little bit of noise. And so this is sort of the diffusion paradigm. It works quite well, but of course it's not perfect because well SDs are sort of annoying to integrate. It may take a lot of steps, so there's been a lot of work on how to speed these things up. So the next thing is sort of continuous normalizing flows. So these were sort of the most popular thing for a year or so before diffusion really came and made it much easier to train. So they sort of fell out of fashion because they were difficult to train, but more recently they've come back because, well, we finally figured out how to train them sort of quickly. And so here we instead of adding noise, instead of having a noise parameter, we just learn sort of directly a vector, right? Directly parameter is an ODE that goes from noise to data and then reverse it from data to noise, basically. So yeah, this sort of is the easier way, but of course, and faster to integrate, but you sort of, it was hard to train. So now we're going to talk about sort of how you train these with flow matching. Yeah, so okay, so I guess how I think about it is you take your sort of data distribution and you're noising it. So you sort of define a series of probability distributions over time. So here it's row 0 to row 1, and you sort of get interpolation between the two. And then your generation is to interpolate back. So you want to learn a function basically that transforms your density over time to either go from data to noise and then vice versa from noise to data. So how do we do that? Well, so we've got to learn sort of a vector field that says basically how the distribution changes over time. So this is going to be this UT, and so that says for any point in time and space, which direction should I go, sort of to change the probability distribution over time to match this row 0 to row t. And so, right, there's sort of a U that generates a row, and that's the one that basically, if you follow that vector field over time with all your particles, then you will match the density over time. And so really, we want to learn this thing, and that'll be parameterized by a neural network that says, at every point in space and time, here's the direction I go to sort of make my noise go to data or data go to noise, vice versa. And so what's nice about this is basically we can do a simulation-free objective, which I'll show shortly. Cool. Okay, so basically learning this UT directly is very hard, but it turns out there's sort of a formula that's conditional. So just like sort of how in diffusion, it was really hard to learn the true score, but much easier to learn the conditional score. In conditional flow matching, it's sort of the same way. It's basically you're able to condition on data on both sides and then learn sort of an easier vector field. So this is showing basically random pairing. So sample a data point from both sides, right? Here it's red, here it's blue, and then sort of at each point in time and space, then regress the direction to be pointed along these lines. And so if you do this procedure, then you'll learn a vector field that sort of nicely moves these data points from one distribution to another. It's not going to be the same conditional distribution, but it is going to be the same marginal distribution, which means that basically once you take this whole distribution, it'll move over time to the correct sort of from row 0 to row 1 or vice versa. And so yeah, this is sort of a random sampling, but what's interesting is sort of optimal transport sampling. And so we'll talk about why that's sort of an interesting thing. But yeah, this function is much easier to learn if you condition on 2 data points. So sort of the vector field that generates the distribution that goes to from a sort of small Gaussian around one data point to a small Gaussian around another is just a straight line basically. And so that's easy to compute. And so once we have this easy to compute thing, we've sampled, and then we're able to directly regress, and that's a very easy loss to learn. And cool. So this G is sort of the metric space, and so oftentimes we do Euclidean space, but of course in proteins we also have to do rotation space. So depending on the sort of the norm here, it may be more difficult to calculate or easier to calculate. Cool. So yeah, so this optimal transport coupling. So what does this actually look like in practice? So here's an example where we're going from 8 Gaussians to 2 moons, and this is on the left is basically if you take random samplings, here are the paths that you learn in yellow. So these paths move from the 8 Gaussians to 2 moons, and yes, in fact, it does do that. However, they're very sort of circuitous paths, right? Like there's many different ways to go here, and they're definitely not the shortest. But it turns out if you do the optimal transport ones, then this is what you get. So this is performed with mini batch optimal transport. You sort of go directly there. And why do you want to do this? Well, it's really fast inference, basically. So the sort of straighter your lines are, then the faster inference you can do because you can do sort of fewer steps for the same tolerance. So for instance, if the line was completely straight, then you could do it in 1 step. So sort of by reducing the variance of your objective and sort of making that path straighter, then you can get faster sampling. So this is sort of one of the tricks we use in FoldFlow to get faster inference. And in practice, this needs maybe sort of maybe a tenth of the steps really to work just as well. So this is a very useful trick, I would say. Cool. And so I'd also said at the beginning, sort of easier to implement, right? So what does this mean? So SE3, right, of course, was space of translations and rotations. So translations has always been easy because we've always been able to do sort of the score in Euclidean space is very easy, but the score in SO3 space is very hard, right? So the score is sort of how heat diffuses over time. How heat diffuses over time in rotation space is dictated by this isotropic Gaussian. And so basically the sort of diffusion models need this the score of this distribution. And that's sort of a very difficult thing to do, right? It's an infinite sum, although it does converge quickly. It can also lead to a lot of bugs. But sort of the flow models don't need this, right? You only need sort of the flow from one point to another rather than how heat diffuses over time. And so this means that we can sort of do a very simple formula, analytic sort of closed thing, and it's much faster and easier to implement. So this, I think, is sort of more numerically stable also. So this is sort of a nice trick is that sort of heat diffusion in manifolds is often very hard, but of course straight lines and geodesics on manifolds is much easier. So we think sort of flows on manifolds are much easier to do with flows than with diffusion. Cool. Yeah, and so final thing is sort of putting this together for FoldFlow. And so I haven't talked at all about architecture, but just about sort of how this works, right? So basically FoldFlow is going to parameterize a prediction of your rotations and translations at time 0. And it's going to have a loss that says, well, here's sort of our parameterized prediction of R0 and X1, sorry, R0 and X0. And you want to match the sort of velocity over time. So in this case, right, it's sort of the change in RT, the conditional change in RT, and the conditional change in XT. And so this is the objective is basically you sample a random place in time. You do optimal transport between your sort of rotations and translations. And then you sort of sample along your conditional field, which in practice is just along this line. And then you do this loss that says match your velocity to the expected velocity, sort of expected conditional velocity at each time. So yeah, this is all you have to do basically. This loss is sort of the main thing. There are many auxiliary losses that can be used and in fact are helpful, but this is sort of the simple formula. So yeah, I guess any questions at this point before we move on to FoldFlow 2? Let's see. Can I see questions? Perfect. Nope. Okay, cool. And so yeah, Guillaume will talk a little bit about FoldFlow 2 there.
Okay. Yeah, thanks, Alex. Okay, so now that we've introduced the losses and I guess the background in theory a bit more, we'll present the newer model FoldFlow 2. Okay, so yeah, the main idea in FoldFlow 2 was to do conditioning on the structures to add more information or representation coming from protein. And we thought like sequence conditioning is a natural way to start. And so generative model in general need to be guided to do useful things. So currently we can guide them with inpainting, potential. For example, in RFDiffusion, they have like these loss function that they use the gradient of during inference. Which works well, but without like using the protein sequence would be a great way to guide it. So protein sequence are highly informative about the structure, and this was shown, for example, in ESM 1 and 2, but also like there is much more data available for these things, so we can train larger model or use very large model, pre-trained model. So yeah, therefore we thought it was a good idea to start with sequence conditioning, and in fact, other research lab have worked in this direction as well. Okay, so by incorporating the sequence and structure, we gain a lot of capabilities in terms of tasks we can consider. So for example, the first task we can consider is the same as in FoldFlow. The previous FoldFlow or RFDiffusion is the unconditional generation. So now we would start from a fully masked sequence and start from only noise as the structure and learn to generate structure like this. Folding, in this case, we would start from a known sequence, so unmasked sequence. A structure would be a noise, and we would learn to create a structure from this given sequence. Oops. Okay. We can also do like inpainting or motif scaffolding. So now we have like partial sequence, partial structure, and we design the entire structure. And binder design, where we would mask part of the chains. Some chain will be masked and some structure of chains will be masked. And then we create these binder in this case. And another task interesting is the partial folding, where we would start from let's say we mask a sequence and unmask the target sequence, and we instead fold the target and design the binder, for example. So we fold and design at the same time. So how to do this, we'll present in the architecture of FoldFlow 2. So yeah, we'll follow the overall pipeline is follow like some similar architecture. The three main stage are these encoder, the processing of the representation, and then decoding to get structure representation. It's similar to existing folding model, except now we have like this structure encoder. And in this case, we also don't use any recycle, but we will talk about this a bit later. So yeah, in the following slide, I'll go over the main components of each block. And not shown here is that there's like a lot of layer norms and little things like this to mainly scale the different representation we have throughout the pipeline. Okay, so to start with the structure and sequence encoder. The structure encoder is this invariant point attention or IPA transformer. It's used, for example, in FrameFlow and other work. And then the sequence encoder is a mix of ESM2 and some like learnable pooling across the layers of ESM2. So with each of these encoder, we get a single and pair representation. We akin to like having a edge and node embedding, for example. The IPA also gives us a rigid, so like the frame, which we'll use then to update our atom position and other structural representation. Okay, so now the trunk, this is like the middle part where we combine all the different modalities. This is a simple like combiner first, where we project all our representations, so single and pair, and then concatenate them. We then feed this into the ESM fold variant of evoformer. In our case, we only had like 2 blocks, but we tried with a few more. And oops, sorry. And yeah, so the combiner module is really just linear layers and concatenation and layer norms. Okay, so after that, we get like one unified single representation and one unified pair representation, where now it's really a mix of sequences and structures. And the decoder, it's again some IPA transformer. However, in this case, we only use the rigid representation, so like the frame representation. And with this representation, we compute the vector field in order to compute our losses and other auxiliary losses. With this rigid representation, we can also have like the atom, the 4 atoms, 3D coordinate and things like this. We found like that the adding skip connection between the encoder and decoder were very useful because when we construct the first node embedding, there's also an embedding for the time, and it was very important to pass back this representation of time in the network. Yeah, as we learn a vector field, like we know that time is quite important. And so at inference, how this model works, we would do some, so it's, as Alex said, we need to discretize an ODE during inference, and in FoldFlow, it's we use like an Euler integration to discretize this ODE. And so another inference, we will start with the given, so either masked or unmasked sequence, initially a noisy structure that we then feed this representation into our network, get the vector field and do one step in this direction, which updates our representation of the rigid, and we go on like this. So inference is very fast, even though we have a much larger model here because we can cache ESM2 representation, so only compute it once at the beginning of the integration process, and then you reuse the same representation throughout the ODE. So yeah. Yeah, so I'll just go briefly over training. So we'll first draw some training example, extract some rigid features, so do the invariant representation that we've presented at the beginning. We then sample uniformly or given a schedule, a time t, and a noise sample x1. So this x1 is a mix of Gaussian and XO sample, so like random distribution on the space of translation and rotation. And then we interpolate between x0 and x1 to define xt, the noisy version of x0. And with this, we define our true vector field ut. We then get the corresponding sequence to this protein. We either mask it entirely or keep it unmasked with a probability of 50%. We pass these two modalities in FoldFlow and get the output vector field like this. And then we compute our losses as explained previously on SC3, compute the gradient and background. So the losses and auxiliary losses are the same as FoldFlow. They include some backbone losses and distance matrix losses with the vector field losses. In this case, we mask 50% of the time. We mask the full sequence 50% of the time. And so this kind of means that half the time we're doing essentially folding. So we can test this a bit later on. And it's also a way to like incorporate the ESM2 representation in the rest of the network. In this work, we didn't do like partial sequence masking. But I know other groups have been doing this, and it works pretty well. So it's something we'll probably do in the future or look into. Okay, so now in the paper and in general, we're interested in other tasks like motif scaffolding, and so we add a fine-tuning specific for motif scaffolding. So on the GIF, you can see like the motif scaffolding is the motif part is fixed, and then the scaffold is the thing in blue that we want to design. And so what we had to do, we fine-tune, we create these like random masks to create some data augmentation on the existing PDB we had. And so these random masks, they define some motif and scaffold we want to use. So from a protein, we generate the motif and scaffold randomly again. We mask these inputs. So when we say we mask these inputs, it's really, we say the motif will be fixed, and we only want to design the scaffold. Then we pass this into FoldFlow and predict the vector field. So only updating the vector field for the scaffold part. Compute the loss only with respect to the motif. And so in our example, we fine-tune for over 300 case steps. But other model, what they do is they pre-train with a task like this. So for example, in RFDiffusion, they did this 80% of the time. Yeah. And I think Genie too also do it a lot. The tricky part here was really to do the correct centering at inference and training, where we want to center the motif and create noise around it. Okay, so maybe a quick recap. At the beginning, Alex presented like this first generation model of ML for protein, and they were either like for different modalities or generation for the same modalities. And now what we see more and more is combining multiple modalities for generation and for specific tasks. So recently with AlphaFold 3, Rosetta Fold, Allotom, we've seen mixing the sequence, molecules, and DNA representation to create structure. Also, so in FoldFlow 2, Genie 2, we use the sequence and the structure to generate structure. And recently ESM3 and multi-flow, where now they generate on multiple modalities. So for example, sequence structure and generate sequence structure, and ESM3 functionalities and things like this. So yeah, this is where what's happening more recently. So now we'll present the experiment. We did some unconditional generation and scaffolding generation. We'll present, these are the two main ones. I'll let Alex present the unconditional.
Yeah, so I guess the first thing is about how do you measure unconditional generation? And so right, our model sort of is this first part, and you take noise and then you get a structure. Of course, how do you tell if the structure is good? Well, so this is the sort of procedure to do it. So you, for us, we're generating 50 proteins at different lengths from 100 to 300. That's sort of the lengths we're interested in. And then we apply protein MPN to get 8 sequences for each structure. And then we refill this ESM fold and take sort of the top structure, the best one. And so if any of them have sort of a sort of C alpha RMSD score of less than 2, then we say this is designable. And so basically, why is this a good measure? It's basically if your structure, if you can find a sequence that refolds into it, we think this is sort of a realizable structure, right? Like it could actually be made in real life. And so this is sort of a computational metric to say, are these structures potentially, can you make them? And so this is sort of the first measure, the most important measure, I would say. So sort of the minimum is basically you need to be designable. And so, of course, once you're designable, well, what else do you want to look for? You want to look for sort of how diverse you are. So in this case, we measure diversity in a couple of ways, but the main way is basically looking at sort of the average pairwise TM score of the designable proteins. And so basically, how different are the proteins from each other that we generate. And the second way is sort of looking at clustering. So looking at how many different unique clusters you get for the proteins. So that's the sort of diversity part. And the second part is sort of, well, how new are they? So if you have sort of a reference set, then you want to look at, well, how far away are you from the reference set? And so novel designs are basically, are you far enough away from the training set? And so this sort of metric is TM score, basically. So you take the distance to your sort of closest element to the training set for all generative ones. And if it's sort of far enough away, then it's novel. So in our case, we chose sort of 0.3 as a sort of fairly novel metric sort of threshold. But yeah, you can choose many different measures here. So yeah, that's sort of the idea is basically you want to move your generated set sort of further away from your reference set, although this is sort of a careful balance because, well, you want to generate your data, but you also want to be able to generate new things. Cool. Okay, so yeah, so okay, basically we measure these sort of percentage of designable, percentage of novel, and percentage and sort of diversity of the different models. And basically we see, well, FoldFlow does quite well. FoldFlow 2, sorry, does quite well on all these metrics. And yeah, I guess the why is sort of, well, there's a couple different changes that we made, and I think there's a couple different pieces that contribute to this. It's sort of adding sequence data really helps. So it's very interesting, right? This is an unconditional task, so we don't have any sequence data. But even without the sequence data, basically helping during training, having the sequence data seems to help the representation. So sort of this says basically you're able to train a better unconditional model, which has no input of sequence. But sort of using previous sequence data seems to help the representation. The final thing, we sort of added this other piece, which is the evoformer. So it's sort of a more powerful representation, so a little bit slower, but it seems to sort of be a good trade-off to sort of add more parameters to your model. And this allows us sort of to get better unconditional sort of both in designability, but also sort of these novel and diverse tasks. Yeah, so one interesting thing we found. So there's always been this trick, basically. Okay, so in sort of the most recent flow matching models for proteins, there's this inference annealing, right? So this is on the right. I guess you can't see my cursor, but sort of the inference annealing trick is basically you want to speed up the rotations relative to translation. So when you're doing inference, right, you would think, well, we do sort of this training where we sort of noise things both at the same time. But during inference, it turns out that it's much better to speed up the rotations relative to the translations. And so this basically means you should determine your rotations first and then learn your translations based on that. And so that's actually what's happening in these models. We're all using this trick that basically speeds up the rotations and then determines the translations based on the rotations. And so you can see sort of on the right, basically we've always picked this sort of 10 times time. So time is 1 at noise time and 0 at time at data. And so it's sort of very fast in the beginning. And then as you get closer to data, it slows down the rotations. And so this seems to be a fairly good trade-off. And so basically, depending on your different choices for here, you can get more designable proteins. And so that's sort of a trade-off is basically you need to do this rotations first and then translations to get really designable generations. And the other thing is sort of the integration step. So, in theory, the step count shouldn't matter. You should get, once you have enough steps, then it should be sort of all fine, right? But in practice, we see a trade-off where you actually, if you do fewer steps, you make it more designable proteins. And we think this sort of interaction with the inference annealing, because we sort of messed up the inference procedure to get better designability. So we sort of do fewer steps to get more designable here. but as you can see, it also trades off the novelty. So yeah, you may get sort of less novel things. So it's sort of another parameter to play with. So yeah, we're sort of interested in these different choices, right? Like I think training inference is sort of a theoretical thing that links them together, but of course, in practice, there's sort of many tricks you can play to sort of get better inference in sort of whichever way you might want. So yeah, playing with these parameters is very interesting to try and figure out, well, decoupling the inference strategy from the training strategy, I think is it's sort of you can develop things that help across models basically. So we think this sort of trick helps across models if you want to sort of develop more designable things. And so yeah, I guess the one thing I'll say about this, right, is that it actually relates to sort of diversity and sort of the secondary structure. And so basically this is also related. Why do you get more designable things? Well, there's a couple of reasons. We think it's sort of easy. These models sort of have a preference for it, but also the secondary structure. Basically, there's some secondary structure elements which are much easier to design than others. So in particular, if you have more helices structures, then it's much easier to design than if you have more beta sheets. And so yeah, this is sort of the secondary structure results. And so for different models, basically, everything loves alpha helices. So the more alpha helices you make, alpha helices are much more designable. But in general, right, if you want other sort of structures, they may be less designable because I think, well, it's just hard to design. There's more long range features. And so these models struggle a little bit with this. But FoldFlow 2 seems to be doing, well, at least much better than FoldFlow 1. And when you can do extra fine tuning for diversity, it does even better. And so that's sort of the secondary structure stuff. Cool. So I'll let Guillaume continue on sort of the folding.
Yep. So yeah, during the training, I also mentioned like we masked the sequence 50% of the time, which corresponds to folding. And so to us, it was more of a sanity check to compare if we were indeed like looking at the sequence information. And so our goal was not to be like better than ESM fold and things like that, but just to be like in some sort of ballpark. And so here we see that if we refold this is like a 300 test case, test PDB that were not seen in ESM fold, multi-flow, and FoldFlow 2. So like our RMSD is greater than ESM fold, but smaller than multi-flow. So yeah, this was a good sanity check to visualize the sequence information. But now for our other main experiment was the motif scaffolding. So at the top right here, it's like one of the motif scaffold. In red is the motif, and in blue the generated scaffold. And so we reuse the RFDiffusion benchmark, and I'll go and explain how did we validate these. So we have the motif, the structure and sequence, we give it to FoldFlow 2, and we generate a scaffold. We keep the fixed part corresponding to the motif. We feed this into Proteinium MPNN and refold it with ESM fold. And now we have 3 score, one align RMSD for the entire complex, one align RMSD for the motif part, and one align RMSD for the scaffold part. Each of these have like a different success criterion, and we say that the generated scaffold is designable if all 3 are true. So we reuse the RFDiffusion benchmark consisting of 24 examples with 100 attempts each. And so FoldFlow 2 and RFDiffusion were able to solve all example. However, we should note that like RFDiffusion was much more designable. So for example, we see in some cases we design a few where RFDiffusion is able to design much more a greater number of designable scaffold, but both methods solved all of them. So I didn't like saturate this task. We created a new task, which VHH a nanobody, where we want to design the region outside of the CDR region. So we want to keep the CDR fixed and design the rest of the protein. So with a bit of processing, we end up with like 25 designable structure that we create for a test set, and we add more like in-domain training data for FoldFlow 2 and compare with RFDiffusion. So with a little bit of fine-tuning, so less than 300 steps, we solve 9 out of 25 and RFDiffusion 5 out of 25. So yeah, these are interesting or encouraging results at least. And we finished with like this zero-shot equilibrium conformation. Given that we saw that FoldFlow 2 was generating more diverse structure, we wanted to see if it was able to like reflect the stable distribution of a protein structure, so corresponding to the equilibrium conformation. So from one sequence, we folded multiple structure, generate multiple structure, and look at the variation of the structure. So the ground truth here is from molecular dynamic, so we can see like how each residue move in space, and we compare with ESM flow MD, a model specific for molecular dynamic. And so we see like again able to generate some diverse representation of the same sequence. So this is it for the experiment. And so maybe now for the next step and some takeaways, this new sequence condition model improved designability and diversity. We solved all of RFDiffusion scaffold benchmark and introduced a new scaffolding task. And so some interesting next step is to use FoldFlow 2 in a wet lab experiment, and also to explore more the binder design. And so one experiment we were looking into is to fold the target and design the binder at the same time. So this is the GIF presented on the right, and design binder at the same time. So yeah, that was a big team effort for FoldFlow 1 and FoldFlow 2. And so just would like to thank everyone here, and thank you for the invitation as well. We have more information on the paper, more experiments, and a blog post as well for a different presentation. So now I think we can take questions.
Yeah, great, thanks.
Awesome. Thank you so much for the talk. I guess I'll go ahead and kind of read off some of the questions, but participants can also feel free. I will let you guys unmute yourself if you want to read your own or kind of follow up. So the first question I'm seeing was asking for the interference. Are there other inputs other than the noise?
Yes. So yeah, maybe I can take that. So it depends on the task. When we want to fold, we would give the entire sequence. When we want to do like a binder, we would give the sequence of the target. But otherwise, the structure is noise, except for the scaffolding where the motif will be fixed, but it's sequence and structure representation.
Yeah.
Awesome. The next question asked if you used a FAPE loss.
I don't, yeah, we didn't, but we used some sort of other losses proposed in FrameDiff. And so there's a, sort of, yeah, it's based on distances, but it's not exactly the same.
It's something similar for the position of the atom there.
Yeah.
And then I guess a follow-up to that was, what was the reasoning behind that? I would assume it's just because of the way your architecture is structured and what you're trying to accomplish. But if you guys want to expand on that, I can just comment that to them.
Yeah, I think, I mean, we basically used what people used before. I think there's still many, like, basically, right, you just want a good structure. So as close as possible. So yeah, it could very well be a good idea to use frame out precision, right? I think that's very possible. I don't know the best combination. So we use sort of a combination that's very many parameters, but it could be simplified if you change the losses. So yeah, I think that would be a good idea to try, but we've not done it yet.
Awesome. The next one is a bit long. The FoldFlow 2 paper emphasizes expanding training to synthetic structures from the AlphaFold database. But for the main unconditional generation experiments, the paper writes that training was only done on PDB structures to get good designability. Hence, does it mean that training on the AlphaFold database is not really helpful after all? What are your thoughts? On the other hand, Genie 2, for instance, does train on the AlphaFold database and achieved good designability. So any comments towards that?
Yeah, that's a good question. You want
to? Yeah, so we think it's doable, but the current result we had, it was more designable with the PDB dataset. But we believe with a different filtering procedure, it might really, it should improve like seen as well in Genie 2, and
Yep.
it's something we're still investigating and working on actively.
Basically, I think eventually we need to move design beyond the sort of current designability, right? So this designability depends on both protein MPNN and ESM fold, right? And protein MPNN is only trained on monomers from PDB, right? And so essentially this metric is overfit on PDB. And so, you know, basically we think that if you want to do designability, you should probably just train on PDB and probably even just the structures, right? That that protein MPNN, you know, trained on, right? But that's of course not the goal. And so, you know, but we don't have a better metric now. So we think, yeah, outfold data DB is sort of a good thing to train on, but it's, well, yeah, we're not ready. Basically, we don't have a good metric to measure that yet. So yeah, that's current thoughts, I guess.
A follow-up from the same participant. Why exactly do you think it is that at inference, it is so helpful to change the schedule and generate the rotation first?
Yeah, this is a good question. Yeah, so I think, you know, nobody's done a specific study on this, but it's definitely useful. But the general thing is basically the secondary structure, sorry, the rotations determine what secondary structure element is going to be where, right? So this is essentially, it's much easier to first determine which secondary structure elements are where and then move them into place versus trying to sort of fix the 3D positions and then figure out what the secondary structure elements should be. So it's sort of, I think it's basically doing secondary structure first is really what it enables to do rotations first. Yeah.
Awesome. The next question I have is, to what degree do you think of binder design as simply an in-painting task on multiple chains?
I mean, yeah, I want to say yes, this is exactly this, but that wouldn't be true because, I mean, now that we experience with this a bit more, there's new complication that arise from this. So one of which is, for example, in the positional encoding, you want to make sure like the two chains are well separated in space, but also during training, now you may want to start from a mix of noise and structure, where the structure will be the target. And so where do you position this in space? It's another like decision to make. We think it should be like centered or something like this, but like yeah, I don't know. I feel like in spirit they're very very similar, but you might encounter different challenges while working on this.
Yeah, I mean, I think it's like, yes, conceptually it is, but in practice there's like tricks will help.
Yeah, exactly.
Right? So yes, it's definitely in painting, but as always, there's more things to do.
Awesome. The final question I have as of now is, you wrote in the paper that RFDiffusion makes almost exclusively helices, while FoldFlow 2 also includes other secondary structure types. Do you have an explanation for this difference?
Yeah, that's a good question. I mean, so we don't really know how RFDiffusion was trained, right? And we observe basically that models, like based on length and other things, that like display very different secondary structure characteristics. So FoldFlow, for instance, seems to generate more diverse things on shorter lengths, and RFDiffusion seems to generate more diverse things on longer lengths. And I really couldn't tell you why this happens, right? So I think it's, yeah, I think nobody really has a good recipe for how to generate diverse secondary structures across, you know, sort of lengths and things without fine tuning, right? Like you can always fine tune to get it, but you know, I think the sort of the biases in the model currently make it so that alpha helices are just more prevalent. So sort of fighting this it's sort of unclear the best way to fight this, I guess.
And it feels like helices are easier or more like designable. So if you work on your model trying to improve designability, you might end up.
That's correct. Yeah, exactly.
Awesome. And the same participant also asked if you plan to make the code for FoldFlow 2 available or on a server.
Yes. Yeah. So we're working on this. It's just it's entangled with company code, so we need to disentangle a little bit. Yes.
Awesome. Okay. Well, thank you so much, both of you guys, for being here today. And we will have our next set of speakers out very soon to the community. So yeah, thank you guys both for talking about FoldFlow 2 today. We appreciate it.
Thanks so much for organizing. This is
Thank
great.
you.
Yeah.